Hey guys, it's been 3 days since I originally swabbed my swabbing plate and left the other plate open to the air for 30 minutes. So this could take 2 to 5 days depending on the temperature in your residence, things like that. So you want to really check it after 2 days, 3 days, etc. But I'm putting mine off at 3 days because I got pretty cool results. So what you're going to want to do for analyzing your results is take a look at your plate and see if there's any distinct colonies. If so, count them, describe them. On the air plate, you're probably going to see a mixture of fungal and bacterial colonies. So fungal colonies tend to be kind of fuzzier. They often are larger, but very fuzzy kinds of things. Bacterial colonies tend to be almost perfect circles and very distinct. So one little bacterial colony has millions of bacteria in it. So let's see what grew off of your fomite, off the surface that you swabbed. Don't forget, you need to be wearing your complete PPE at all times during this, even the second part of the lab where you're analyzing your results. There's some nasties in here. You do not want to open these Ziploc bags, okay? Do not open these. And then we're going to show you proper disposal techniques of what to do after you are done. So take a look at it. I'm going to show you the air one I got first. I hope this comes up in the camera. My petri dish open to the air for 30 minutes. You can see there's some interesting things that we're breathing in at all times, but fortunately most of us have an immune system that guards us from these things affecting us too badly. But anyway, so that's my one from the air. My one from the from what I swabbed, and again I still swab my cell phone, and I got a mix here between distinct colonies. See if you can see anything that I'm looking at here. Mine came out all white. Depending on the species of bacteria, they might come out a different color. But you can see mine, there are very few distinct colonies. There are some, but mostly I got tons of growth. I just got complete kind of carpet of growth there where I swabbed. I don't know if you can see that. Oh, there you go. Okay, if I change the light a little, it seems to work better. See all that white stuff over here? That's all tons and tons, probably trillions of bacteria right there. Everywhere where there's a distinct colony, a distinct little thing, there's millions of bacteria. So in this case, I can count a few of these colonies. The distinct little dots, I'd say 1, 2, 3, 4, 5, 6, 7, 8. I probably have maybe 9, 10. On the whole plate, probably 10 distinct colonies. But the rest of it is just a carpet of bacteria, and you can't distinguish on mine individual colonies. In other words, let's just say there is a whole lot of bacteria that was growing on my cell phone. Nasty, huh? Now, just imagine what's growing on the bottom of your shoe. So if you walk into your house with shoes on, you're grabbing all that bacteria and walking it all over your floor of your house. Anyway, with that happy thought, again, I will try to take pictures of this stuff with my cell phone, but I'm going to pause this to take the pictures, and then I'm going to come back and show you how to safely dispose of what we have here, because you do not want this getting out in your house. So hang on one sec. Okay, so I finished taking pictures with my cell phone of my 2 plates, and I'll share those with you at the end here. But what I want to do right now is show you how to properly dispose of your plates. So what you're going to need to do, again, complete PPE, including gloves. You're going to need some household bleach, Clorox bleach, and we're going to be making a 5 to 10% solution, bleach solution, in which to disinfect everything that we've worked around and to also store the plates in. So first thing you should do is make up a 10% bleach solution approximately. Get some kind of like a Rubbermaid container. It should have a lid with it, like a plastic container with a lid. And you're going to want to, I've already made my bleach solution here. So basically to do this, you have to kind of eyeball it in this case, but you want to basically add about 1/10 of bleach. And so to do this, you want to fill first the bleach to about right there, and then fill the rest of the way with water. So a 5 to 10% bleach solution. From there to bleach, and then the rest of the way water. And then what we're going to do is we're just going to place our petri dishes with the bag inside the bleach solution, and just let them soak there overnight. So for about 24 hours. And then once you've got it in there, you can slightly open the bag just a wee bit to let the water in so it sinks down to the bottom. Put the lid on and let that soak somewhere safe, like in your garage, for 24 hours. And then you'll be taking those petri dishes and bags out of the bleach solution, immediately put them into 2 layers of trash bags and tie them up really good, and then throw them out in the trash, and you should be good to go. You also want to get a spray bottle, something like a regular household spray bottle from Home Depot. Make sure it's nothing that's ever had an ammonia-based solution in it, so no Windex, no Lysol, anything like that. That's ammonia-based because bleach and ammonia create chlorine gas, was not a good thing. So, but get a empty spray bottle that has not had an ammonia-based solution in it, and make a 10% bleach solution with that. So again, 10% bleach, the rest water. So 1/10 of it should be bleach, the rest 9/10 of it water. And just go ahead and spray anywhere where you've been handling these petri dishes inside of the Ziploc bags. And that will just let that dry. You can take a paper towel to smear it, but then let it air dry, and that will disinfect the surfaces. Okay, I look forward to seeing your pictures. Don't forget the pictures. Look forward to seeing that on the discussion board. And okay, here's some photographs of my plates, just to kind of point out to you what we're looking at. So this plate over here was the one that was left open to the air. And if you look real closely, you see things like this. Like, see that little guy and that little guy? See how those are perfectly round and very tight? Those are bacterial colonies right there. Whereas something like this, you can see how it's kind of feathered around the edges and it's a little bit larger. That is most likely a fungal colony. Same with this one. You can see how it's big and feathered. So you'll see on the one left open to the air, you'll probably see a mix of bacteria and fungi. Whereas you come down here, this is the petri dish that I swabbed my cell phone and then inoculated the dish. And what you can see here, so this is all bacteria, but here you can see distinct colonies. So you can actually count those colonies, like 1, 2, 3, 4, 5, etc. You can come over here, 6, 7. Whereas up here, there was just so much bacteria clumped up that they did not actually isolate. It didn't separate. So there's just tons and tons and tons of bacteria all over my cell phone there. And you can see the ones here are actually a little yellowish. So those are possibly a different species, like Staphylococcus aureus can give kind of a golden color. Anyway, so that's just how to see your plates, and just try to describe them in your handout. Anyway, hope you had fun doing this activity. You at least learned some simple microbiological techniques. Just be safe and make sure you dispose of it properly. Take care.